Thank you. So I'm Jess McDonough, co-founder, COO of Attivare Therapeutics, and I would like to thank the conference organizers for having me here today. It's quite an honor for us. And today I'll be talking about Addimune, which is an innovative modular therapeutic approach to cancer immunotherapy being developed by Attivare Therapeutics. So to get started, Attivare is an innovative tech bio company. We're a spin-out of Harvard University. We started in 2021, and we're preclinical stage. We're really interested in targeting patients with high unmet needs, particularly patients that are refractory to immune checkpoint inhibitors. We're a venture-backed company. We've raised a VC seed round in 2021, and then we also have non-dilutive funding support from the Gates Foundation for a separate infectious vaccine disease program. And the nice thing about that funding is the core biomaterial technology is common between the oncology as well as the vaccine side of the business. So what's the challenge that we're trying to solve here? And I think it's what brings many of you here today, is that most patients won't respond to immuno-oncology drugs. And it's estimated that if every cancer patient late stage was eligible for Keytruda, that only 12.5% of those patients would respond. In clinical trials in combination in late-stage patients, the overall response rate can get pushed up 20 to 40% in combination, depending on the specific cancer, the stage, the molecular makeup. But there's still several challenges, including tumor heterogeneity, which I know has been mentioned, an immunosuppressive tumor microenvironment, which is really something that we're very interested in exploring at Attivare, and tumor evolution, resistance, that emerges over time. On the drug design side, it's also very challenging because several of the drugs that are available have issues with systemic exposure and high toxicity. So getting the right dose to the patient is very, very challenging. In addition, where innovation has gone is becoming increasingly complex with cell and gene therapy. And so you end up with these really long development timelines and complex drugs to solve some of these challenges. And it's increasingly challenging to show improvement of combination therapy. And so for incremental increases in efficacy, you also get an increase in toxicity, which makes it very hard to manage. So at Attivare, our goal is to build a personalized oncology immunotherapy platform. And this is enabled by the core biomaterial technology that we've licensed from the Wyss Institute at Harvard. And so the goal is to basically have a patient come in, ultimately, get a tumor biopsy, get transcriptomic data, have an AI algorithm look at that data, group it with like patients, predict which agents would best respond for those patients, and then basically formulate a custom pharmaceutical product at the hospital pharmacy. And the goal there is really to have the biomaterial scaffold available, and then different GMP immune agonist options. And then with a simple aqueous admix, about 30 minutes, a patient could receive a customized therapy. And then following those patients and biopsying, if available, or following ctDNA over time to treat those patients. This is the long-term vision, and we're taking a practical stepwise approach, which I'll walk you through, which the Oncode Accelerator will be a part of. So how are we building out this vision? We're basically thinking of our core biomaterial technology as similar to a hardware. We think of it as like the computer, and then the immune agonists that we load onto it as the software to program the immune system. And then we also are layering in an AI compute layer as well. And so we're working with a biomaterial called mesoporous silica rods. These are silica. They're 100 microns long, 4 to 6 microns across, porous throughout. And what that means is essentially they're much bigger than cells. They're almost like splinters in the tumor. And so when you inject those into a tumor microenvironment, they sit in the tumor like a bird's nest. One of the nice properties of silica is it absorbs many compounds without any chemical modification. You might have seen silica packets and different things, filtration uses around. And we're taking advantage of this property to develop modular drugs. And the idea is the silica basically stays in the tumor for up to 3 to 4 weeks, which solves many of the PK/PD issues of intratumoral drugs. And then again, it enables this fast 30-minute hospital admix. The next layer, the AI compute approach, is really using targeted patient data with transcriptomics. And there's been incredible innovations on this side as well, with AI algorithms actually predicting how patients will respond in clinical trials, and then it being concordant with the actual clinical results. And then basically building out an algorithm that could predict which immune drugs would best serve that patient if delivered locally in the TME. And then finally, the immune agonist software side, there's several options, lots of different immune modulators to choose from here, but they're very challenging to dose systemically. We've worked a lot with interleukin-12, which is a very potent anti-tumor cytokine, but very toxic if you give it systemically, and very effective if you can give it locally at the right dose. We could also longer-term load neoantigens, viral antigens, to build out a cancer vaccine approach as well. And so basically, by combining these three layers, the idea is to have the silica rods, the drug product, mix, inject into the tumor microenvironment, have that interaction, attract in the immune cells, inflame the tumor, and get a systemic anti-tumor response. And so why does hardware, software, and compute matters? As we've been talking about, systemic exposure is really associated with off-target toxicity, so this helps solve for that problem. It's also very difficult to combine assets because of those issues with toxicity and PK/PD. But if you put them locally at the tumor microenvironment, it again solves for some of those problems. And slow to test, years per iteration, where basically for us to make new drug compounds on the rods, it only takes the amount of time for us to order the compounds. And no personalization versus a personalized mix and match approach. And so this is our lab. This is our AI fab, our ATT AI fab, where we make the silica rods. And you can see it here. The MSR technology is based on about 10 years of publications from David Mooney's lab, who's an expert in biomaterial technology, and done several projects around programmable release, immune cell recruitment. In the company, we've done a lot of work on complete in vivo degradation to make sure it's safe, it stays where we want it, biodistribution, and it's also very compatible with immune agonists. We've also built out the platform to show the hardware reproducibility with partners, built out the clean room, SOPs. We have a quality system established, and we'll be at GLP, GMP next year for the biomaterial component. And here's an example of the first software program, which is interleukin-12 on the MSR rods. These are syngeneic murine models. And so at the top, you can see single tumor models, where basically we're injecting either saline, in green, interleukin-12 alone, or the MSR IL-12. And you can see in the colorectal melanoma model and triple-negative breast cancer model, we get really nice results with complete regressions after only a single treatment. And this is not in combination with anything else. And then what got us really excited was the abscopal two-tumor model, where basically you take a mouse, put a tumor on each side, inject one tumor one time, and we saw really nice effects on the other tumor as well, including complete regression in some mice, as well as the similar gene expression profiles on both sides. And then the other piece, too, is, again, low systemic tox. And so here we've done an experiment in mice where we basically injected IL-12 versus IL-12 MSR and shown that the IL-12 alone, you get that bolus in the sera, but with the MSR versions, you don't see it at all. And finally, we've done gene expression studies a week after treatment, and we see all the gene expression patterns you would expect with an interleukin-12 treatment, including the IL-12 interferon gamma signatures, T cells, NK cells. And then interestingly, we also see an effect of the silica rods themselves, which have some innate immune activating capacity. And so our goal is to apply this first, and we're very interested in colorectal cancer and primary tumor, as well as METs to the liver, which are an incredible unmet need. And so right now, I'm very interested in MSS colorectal cancer, which does not respond to immunotherapy. And the rationale here is several of the factors in the TME, the low T cell infiltration, MHC downregulation, myeloid dominant phenotype, are phenotypes that we saw reverted in our treated models. We also have access to a data set for CRC. And this is really where the Oncode Accelerator Project comes in, because we've generated some really robust mouse tumor model. We have the murine data, we have mouse cell lines, but in order to bridge to the clinic, we really wanted to get some human-centric approach. And so for our collaboration, we're planning to work with MIMETAS and Amsterdam UMC. On the MIMETAS side, the goal is to use the CRC organ on a chip, use it with M2 polarized macrophages, and then flow PBMCs by that as well, and then treat with the silica alone, silica IL-12, and IL-12 alone and look at the different immune effects. And this will allow us to de-risk the platform, get more information about the mechanism of action, which has been a key question of the investors, particularly in light around the silica inflammatory findings. And then with Amsterdam UMC, they have access to lots of patient samples. And so the idea here is to do in vitro cell culture experiments with primary cell lines and primary tumor cells with the MSR IL-12, and basically look at the effects of the silica, the IL-12, and then the components together, which again helps us build out that robust data pipeline on patient-specific samples, which will help us better plan for our clinical trial, which patients to target, and have a better chance at clinical success. And so we have several competitive advantages here, and I think I've walked through several with the modularity, the AI tied directly to our platform, as well as this tunable recruitment. And we think the modularity combined with just a very simple system will help us win. And then I often get questions about regulatory and clinical plans, because this is building to a complex future. And I think we're starting in a simple, practical approach, plan to file drug master file with the FDA around the biomaterial itself, and then build out that portfolio with different assets. In addition, the AI portion will be consistent across all of the programs. And then we'll start with the MSR IL-12, which is a project we'll de-risk with the Oncode Accelerator in the clinic to show the safety and efficacy of the silica-based approach in a standard phase 1 clinical trial, but with longer terms to build out into adaptive clinical trials. And after some additional meetings with the project partners yesterday, lots of ideas of how to accelerate this with the organoid platform as well as the patient samples. And so the long-term vision is really to build out this data flywheel where we would start to treat patients, collect data, and then over time get faster and faster and better at building out our pipeline of drugs. And I'd like to thank the Attivare team as well as our advisors, and also really like to acknowledge the Oncode Accelerator team, who's been incredible to work with, had amazing meetings yesterday with the project partners, and am even more excited to work with them than I already was, as well as the Attivare team who did this work. Thank you.